Okay, thank you. Yeah, otherwise I was going to request a blackboard. Right? Okay, so I want to talk about this library I've been developing for a decade, and recently I got the addition of 2 new group members, Eduardo Sarti here and an engineer, Com Le Breton at INRIA in Sophia Antipolis. And so I'm going to talk about this library. So to get into the mood, I would like to discuss briefly challenges in modeling biomolecules, because we heard a lot, in particular within 3D-Sig, about deep learning, AlphaFold, etc. 2/3 of the talks were about machine learning, deep learning for molecules, biomolecules, but this was essentially in the static realm, modeling static structures. And so I would like to discuss briefly dynamics. And then I'll be presenting 2 complicated tasks we have solved somehow using our library. And so this will give me the opportunity to mention some of the software tools which are available. And to conclude, I'll talk about the library itself. So, of course, if we are addressing challenges in modeling biomolecules, everybody knows this one, which has essentially been solved. You get a sequence, you want a plausible structure, namely the coordinates of the atoms. But if you're using all these beautiful tools based on deep learning, so there are a number of caveats, which are, you get one structure, essentially. There is a bias towards what has been learned from databases, and in particular, you hardly get any information about flexible regions, right? Or they are not so much reliable, and in particular for flexible regions, a single structure does not really make sense anyway. Of course, you don't have any information about thermodynamics or kinetics. And of course, last but possibly not the least, predicting is not explaining, and so you don't exactly know what's happening. And so we got to talk within 3D-Sig. So does AlphaFold understand biophysics in the first place? Okay, so this challenge is essentially solved, but there are a number of limitations. And now, if we talk about the challenge of dynamics, okay, so where is page down here? Oh, it doesn't work anymore. I'm sorry.
Yeah. Because you clicked on the actual slide, and you can make progress down the road. Click outside of the slide and move your, I think that was it.
Outside? Okay. No.
So Beamer and LaTeX and Beamer is forbidden now.
Okay.
Oh, okay. Yeah, so we have a movie which we won't see here. So this slide is to talk about dynamics, where we want to predict, let's say, thermodynamics, maybe in terms of KD, dissociation free energies, if you care for interactions, which has an enthalpy component, delta H, and a minus T delta H, which is embarking the change in entropy upon binding. Or maybe you care for kinetics, K on, K off, right? And the difficulty here is that the biologically relevant timescales are typically beyond milliseconds. And of course, if you want to study these phenomena using classical methods such as molecular dynamics, integration time steps are typically about femtoseconds. And so it takes, of course, a huge number of time steps in order to achieve the information, to obtain the information you have. And so if you don't know this movie by John Codera and others, I encourage you to take a look to it. It's very nice because it's a simulation which, well, was obtained after 230 years of GPU calculations. And what you see is a protein of moderate size, 1 in 64 amino acids undergoing conformational changes. And at the same time, you get a Markov state model with one node for each metastable state. And you see the protein jumping from one metastable state to another one. And the difficulty here, of course, is that you cannot do this in a routine fashion because this is too costly. And so, in fact, if we want to care for dynamics, we have to jump to the realm of potential energy landscapes where we have, let's say, charm armor, which gives you a potential energy. And of course, as you possibly know, there is a number of critical points, local minima, saddle points, etc., which is exponential in the number of degrees of freedom. And by aggregating these basins, etc., and doing the calculations, you are going to end up with, indeed, kinetic or thermodynamic pieces of information. But of course, as I told you, the complexity of this is currently beyond reach. And so if we step back and in order to motivate what we have been doing, in fact, nowadays, if you care for these properties, in fact, because machine learning and deep learning is so much trendy, in fact, there is a data shortage in order to study dynamics and kinetics using machine learning, deep learning. There are not so many very long simulations around. And secondly, if you care for interaction, this new paradigm, which consists of gluing together amino acid chains and then passing these to AlphaFold, so this, in fact, does not handle dynamics at all. And so my punchline here is that if you care for this information, you have to resort to mixed approaches, which are involving ab initio and data-based approaches. And so what I want to do next is to consider two problems which are related to dynamics and which require a number of complicated algorithms. So the first one is going to be about loops. So as you may know, if you collect in the PDB crystal structure whose resolution is better than 2 angstrom, 80% of these structures have missing segments, and in 90% of the cases, these missing segments are within flexible loops, right? And so here I picked 3 examples of loops. On the left-hand side, you have a homotrimer, which is a membrane protein doing a drug efflux in gram-negative bacteria. And so we have 8 rigid domains which are moved around by 12 flexible linkers, right? And so if you want to understand the efflux, you have to study these dynamics. In the middle, you have CDRs on antibodies, and as you may know, the binding affinity during affinity maturation involves, in fact, the dynamics of side chains. And so if you want to predict a delta GD, in fact, you have to care for dynamics, or you'd better do it. And of course, for IDPs or proteins with IDRs, in fact, it's a mix of folding and binding at the same time because conformational changes typically occur in presence of the partner. Okay, so the first problem about loops I want to talk about briefly is predicting conformational changes of long and flexible loops. So I may call this a move set for protein backbones, right? And so there are a number of tools around available. So they are typically based on the so-called tripeptide loop closure, which was developed long ago by Harold Sheraga and others. Koutsias also, I'm not going to mention the names. These move sets, they play essentially with dihedral angles on the backbone, and they are solving an inverse problem, namely you give me 2 anchor points, and I'm going to consider and find out all compatible geometries with the anchor points for the geometry of the loop which is joining them. The difficulty is that nowadays this method, they essentially work using tripeptides, and in fact, if you have been using Rosetta, the Rosetta kick is also based on TLC. But what was missing around was a move set processing coherently an entire chain. And so this is what we have been able to do. So in fact, we developed a global analytical model for a whole polypeptide chain. And so I'm not going to discuss, it's a bit elaborate, but what you end up with is in fact is a method which is going to generate of the order of 10 to 20 conformations of a long chain per second. And so you get in the middle, for example, the longest CDR3 known to date in human. It's a CDR from an antibody raised against HIV. And as you can see here, for 30 amino acids, we have been able within minutes to generate a huge number of conformers. And so the method itself is, if you're familiar with Hamiltonian Monte Carlo and sampling methods in high-dimensional spaces, this is like a hit-and-run method, a billiard walk in a complicated angular space, which is, in fact, bouncing on curved walls corresponding to constraints on some dihedral angles. And at the end of the day, it makes it possible to reconstruct the whole loop. What is interesting here is that if you want to solve this problem, so let me list a few software packages you have to use in order to do so. Of course, you need to be able to represent a polypeptide chain. You need to manipulate Cartesian and internal coordinates. You need to handle steric clashes. You need to potentially evaluate potential energies using Charmember, whatever you want. You need solvers for the tripeptide loop closure and longer loops. You need to do calculations on group motions, SO3, SE3, rotation, translations. And there are some parametrization involved in angular spaces for the internal geometry of the long loops. And finally, you need to run these complicated MC methods to sample this high-dimensional curve domain. And so, as you can see, this is a complicated endeavor. And so if you want to complete this program, in fact, you have to assemble these pieces of software, these algorithms. And so as far as I know, there are not so many libraries around which make it possible to assemble them. So our library is possibly the only one. So the second problem I want to briefly mention is understanding efflux by membrane proteins. And so here is an example, which is, again, ACRB, this drug efflux machine in gram-negative bacteria. So as I told you, it's a homotrimer with 8 domains, 12 linkers. And in fact, it's using the proton motive force. And so it's a really complicated mechanism. And so you have to understand how these flexible linkers move around the domains which in fact push the drug outside the bacteria. And so here, using a semi-supervised approach, we have been able to identify, in fact, dynamic domains, to assign stable state, metastable states to conformations, and also to identify novel metastable states. And so again, what's interesting here is that in order to complete this program, you need non-trivial methods, which are identify rigid blocks in a protein, possibly related to the chainsaw we heard about two talks before this one. You need to compute molecular distances beyond our plain RMSDs. You need to model interfaces in a hierarchical fashion because, of course, you care for domains and not the entire chain. You need to do some clustering and also complex clusterings. And so again, if you want to assemble all this, it's non-trivial. Which brings me to the library. And so to play a metaphor, let me maybe mention the first image ever of a black hole, which was obtained 4 years back, using images collected by telescopes in 8 observatories. And so in fact, assembling this image has been a complicated task, mixing, of course, data collection and signal processing. And the point I want to raise here is that if you want to do the same to study dynamics, in fact, of course, all proportions kept, you have to handle biochemistry to represent biomolecules, biophysics to encode force fields. You have to handle geometric calculations for surfaces, volumes, coordinates. You have to possibly solve some graph theoretical problems because there are, of course, pieces which are interacting with one another. You need to do statistical physics for entropies, free energies. Machine learning is involved for correlations, dimensional reduction, etc. And of course, if you want to run all this in parallel, you have to resort to complicated architectures. And so, as you can see, in order to do all this, you need a full toolbox beyond gems, isolated gems, which are solving specific problems. And so it's the reason why, like a decade back, I undertook the design of this library, which essentially has 3 columns. So on the left-hand side, you have application for end users. And so this is what you are familiar with as a user. You have a specification. For example, I want to study the interface between 2 molecules, or I want to generate conformations of a loop, a specific problem. And for this problem, you get an executable. On the far right, in blue, in fact, you have low-level complicated algorithms, which are pure computer science applied math methods, which, of course, are generic. They can be applied to molecules, but in many other contexts. And in the middle, you have, in fact, biophysical models for protein, force fields, etc. And because this is C++ with templates, if you plug some class from the middle column into a class from the right-hand side, in fact, you instantiate your C++ class, you end up, in fact, with a program, an executable. So this is a philosophy. And so at a glance in the SBL, we have of the order of 250K lines of C++. So, of course, the caveat is that this is flexible, but it's a bit complicated because if you're familiar with templates, these are like nested Russian dolls, right? You have a class which is parameterized by your class, etc. And so the code design is a bit complicated. And so for developers, there are, let's say, 60 different packages which are ascribed to, let's say, algorithms, geometry, topology, graph theory, optimization, etc., and also to computational structural biology to represent force fields, compute molecular distances, and so on and so forth. For end users, there are like 30 packages with more than 100 executables solving specific problems. There is a huge documentation which we wrote by hand, so many pages. And so we are currently designing plugins for, in fact, PyMol and VMD, both for macOS and Linux, of course. And if you click this link, which I'm not going to do on this computer, there are also a number of interactive demos developed with Eduardo and Nicole. And so I invite you to take a look to, in fact, these resources, the front page of the library, the application for end users, and finally the documentation from which you get a full overview of low-level algorithmic classes. And so to conclude, so if we look backwards, so this has been a major endeavor which I started a decade back, and so overall we have integrated the PhD works of more than 10 students. And so the architecture proved scalable and flexible enough in order to incorporate developments in different directions, because again, it's been really designed to put things at the right place: algorithms, geometry, topology, graph theory, and then model, biophysics, models, proteins, nucleic acid, and so on and so forth. So this major effort has been done, but of course we have not been very successful in terms of users, because we never really advertised the library for a simple reason. We never got resources to fully optimize, in fact, the documentation, the installation procedure. But now things are changing, because we recently were able to hire a full-time engineer. And so sometime in the fall, we'll make a major release advertising, of course, an installation procedure based on Conda, which is already available. So if you just care for, in fact, the application, you have a Conda channel for it. You also, of course, can git clone the source code of the library, but we'll advertise this sometime in the fall. And of course, for the future, there are a number of questions. The first one is to possibly integrate novel methods from the realm of machine learning and deep learning, if such methods make it possible to improve predictions in the realm of dynamics and thermodynamics. Okay. And also another question, of course, would be to integrate community contributions, which is a little bit tricky in our case, because this is not just a Python module, etc. In fact, the design has to be compatible with the spirit of the library in terms of specification by traits class, C++ traits classes, and template parameters. So this requires some careful review process, but of course, we would be happy to do so for non-trivial contributions. And so maybe my final word will be that this is a really significant endeavor, a lot of time. Will it be very successful? We'll see. But as Pascal used to say, the excitement of the gambler is proportional to the expectation of the game. And the expectation, you have the probability to win and the amount of money you will win or success. So we'll see. Thank you.
I should first scan, then I came back to you. Okay.
Yeah, very nice library. Do you have Markov state modeling in this?
Sorry, do I have?
Markov state modeling?
No, we don't. Okay. We don't, but we have, of course, there are the libraries by Pandey and others for this, so we did not. The algorithms which are here are original, in fact. So we did not really, maybe in one or two cases, we replicated something existing, but otherwise, yeah.
Very interesting. I'll take a look at it. I have two questions. The first is if you have within the library normal mode analysis, because obviously it's a low cost and high yield way of exploring.
So we have them. They are not in the release, but we'll release them, I guess. In fact, in the fall, we are going to release normal modes plus the spectrous partitioning algorithm, which is identifying rigid blocks within a protein. And so we'll release this in the fall.
If you include normal modes, take a look at NCOM, which is not sequence agnostic.
Okay.
The second question is related to the loop dynamics that you showed.
Mm-hmm.
Beyond antibodies,
Mm-hmm.
My question about the loop dynamics is the rest of the protein moves also, or is just a loop? Because outside of antibodies, it would not make sense to keep the rest rigid.
So, for example, I think, of course, it's the reason why I put these three examples. Anything happens, right? So in ACRB, this complicated homotrimer, it's a rare membrane protein for which there are like 90 high-resolution crystal structures. And so there, if you take a look to our proteins paper, in fact, we have carefully analyzed the dynamics of domains, and a number of them are essentially rigid, right? So it does happen to have cases where you have essentially rigid domains. And so I don't know about that, right? I have no clue. So, if there is too much dynamics, there is some entropy going on, and whenever you bind two molecules, you pay an entropic penalty. So in terms of free energy, having dynamics, if you don't need them, this is detrimental.
Tell that to the protein, not to me.
No. I'm just making a point about dynamics. Yeah. So